Hello everyone, BiyokimyaVET channel is here with the topic titled Carbohydrates-II. I wish it to be useful to all biochemistry lovers, and please don't forget to like the video to reach more people. Let's start. The contents of Carbohydrates-II are monosaccharide derivatives, amino sugars, amino sugar acids, deoxy sugars, and sugar alcohols, sugar acids, and phosphate esters. Then we continue on disaccharides, Glycosidic Bond, Maltose, Cellobiose, Lactose, Sucrose, and Trehalose. And at the end, Oligosaccharides. Let's start monosaccharide derivatives and focus on these two given monosaccharide derivatives, which here this one is carrying a phosphate group and this carries an amino group. So they are the derivatives. Monosaccharide derivatives, so the organisms contain various hexose derivatives. In addition to simple hexoses such as glucose, galactose, and mannose, there are several sugar derivatives in which a hydroxy group in the parent compound is replaced by another substituent or a carbon atom is oxidized to a carboxyl group. And don't forget here the sugar derivative will mean the monosaccharide derivative. And also remember the substitution reactions from the organic chemistry, and also the redox reactions, again in the organic chemistry. So you may need to check your organic chemistry lecture notes, or you may watch the videos in my channel. So what are the monosaccharide derivatives? Amino sugars, amino sugar acids, actually they are N-acetyl derivative of amino sugars, deoxy sugars, sugar alcohols, and sugar acids. And the first one is amino sugars. Amino sugars are formed when an amino group replaces the alcoholic hydroxy group on the second carbon atom, mostly in monosaccharides. This is a substitution reaction. Amino sugars are named according to the monosaccharide to which the amine group is attached. And for example, if we have a glucose and an amino group attached to glucose, we call it glucosamine. Galactosamine, if it is galactose, or if it is the amino derivative of galactose. And here you see the glucosamine and galactosamine, which are the important amino sugars, which they participate into the structure of heteropolysaccharides, or shortly polysaccharides, such as hyaluronic acid, heparin, chondroitin sulfates, and which are the components of cartilage, bone, tendon, chitin, and cell wall in fungi. So they are universal. And here within these two molecules, as you see, the beta-D-glycosamine having a glucose, we have a glucose carrying an amino group. And it's a substitution reaction because in glucose, actually, in here, we have the hydroxy group, but it is replaced with the amino group. And also in galactosamine, the same thing happens. Beta-D-galactosamine. The second carbon, this is the first, and the second one, and the second carbon is replaced with the amino group. And the glucosamine, also remember the glucose is universal, is the most important or significant molecule of in carbohydrates and for the organisms. And also its derivatives such as glucosamine is part of many structural polymers, and not only for eukaryotic organisms, also for prokaryotic organisms. And the amino sugar acids, most amino acids found in nature are acetylated. In other words, they are N-acetyl derivatives of amino sugars, N-acetyl derivative of amino sugars. That is, they are bonded with acetic acid with the amino group. So if it is glucosamine, it will be N-acetyl glycosamine. Amino sugars and amino sugar acids are often involved in the incorporation of carbohydrate polymers. The physiologically important amino sugar acids are neuraminic acid, sialic acid, and mumaric acid. Here's some important amino sugar acid, and look at the first one, the neuraminic acid, which consists of pyruvic acid and mannose. Neuraminic acid can also be displayed as a product of the aldol condensation of pyruvic acid and D-mannosamine. So, which means 2-amino 2-deoxymannose. Neuraminic acid does not occur naturally, but many of its derivatives are commonly found in animal tissues and bacteria, especially in glycoproteins and gangliosides. And the gangliosides are glycolipids. N or O substitute derivatives of neuraminic acid are collectively known as sialic acids. And the predominant form in mammalian cells being N-acetyl neuraminic acid. This is the predominant form. And sialic acids are a class of alpha keto acid sugars with a 9-carbon backbone. And the term sialic acid, which means saliva from Greek, was firstly coined in 1952 by Gunnar Blix. Then the most common member, as I said, the N-acetyl neuraminic acid, found in animals and some prokaryotes. And N-acetyl neuraminic acid is actually N-acetyl mannosamine derivative found in many glycoproteins and glycolipids on animal cell surfaces. It consists of pyruvic acid too. And the N-acetyl neuraminic acid provides recognition sites by other cells or extracellular carbohydrate binding proteins. And this residue is negatively charged at physiological pH. It carries carboxy group, which is negatively charged donate proton, and is found in complex glycans on mucins and glycoproteins present in the cell membrane. N-acetyl neuraminic acid residues are also found in glycolipids known as gangliosides, an important component of neuronal membranes found in the brain. So, it's very important. Neuraminic acid and its N-acetyl derivative, actually, which is a sialic acid, is so important for organisms. And here is a neuraminic acid, as you see, the pyruvic acid component and the amino group, and N-acetyl, and also it's carboxylic acid, as you see, derivative of mannosamine and pyruvic acid. And here the N-acetyl derivative attached to the amino group of neuraminic acid. And also we have a mumaric acid. Mumaric acid is another amino sugar acid composed of glucosamine and lactic acid. It occurs naturally as N-acetyl muramic acid, or is N-acetyl derivative in peptidoglycan structures, whose primary function is as a structural component of many typical bacterial cell walls. Deoxy sugars are monosaccharide derivatives that usually do not have an oxygen atom at the second or sixth carbon atoms. Substituting a hydrogen for the hydroxyl group at carbon 6 of L-galactose or L-mannose produces L-fucose or L-rhamnose respectively. And L-fucose is found in complex oligosaccharide components of glycoproteins and glycolipids, while L-rhamnose is found in plant polysaccharides. And actually, here is the most important one, or the most biologically significant one, the deoxyribose. And it's a deoxy sugar of ribose. Deoxyribose, or more precisely, 2-deoxyribose, is a deoxy sugar. Its name indicates that it's a deoxy sugar, meaning that it is derived from the sugar ribose by the loss of a hydroxyl group, loss of hydroxyl group. So the carbon-oxygen bond replaces carbon-hydrogen bond. This is a reduction. We need a reductase class of enzyme. It is discovered in 1929 by Fabius Levene, and the deoxyribose is the most notable for its presence in DNA. And remember that the DNA represents the deoxyribonucleic acid, the deoxyribose, it's the deoxyribose prefix. And deoxyribose is produced from ribose 5-phosphate by enzyme called ribonucleotide reductase. And these enzymes catalyze the deoxygenation process. Sugar alcohol. Monosaccharides are formed by the reduction of aldehyde or ketone groups to alcohol groups. And sorbitol is formed from glucose, mannitol from mannose, glycerol from glyceraldehyde. And sugar alcohols are formed by the change of aldehyde groups, forming the first carbon of aldoses to primary alcohol groups. By the change of ketone groups on the second carbon atoms of ketones to secondary alcohol groups. So these are the reduction products of sugars. Let's look at some sugar alcohols and monosaccharides that give the sugar alcohols and their importance. And that's like the sorbitol is a sugar alcohol or the reduction product of glucose and fructose. So sorbitol is a sugar alcohol with a sweet taste that is metabolized slowly. It can be obtained by reduction of glucose, which converts the aldehyde group to the primary alcohol group. And sorbitol 6-phosphate is converted to fructose. So they are phosphated molecules, by the way, during the metabolism. And the pathway is called the polyol pathway, so from glucose to sorbitol and fructose. In the body, glucose reduction is not the main pathway of glucose metabolism. So the polyol pathway is not the main metabolic pathway for glucose. However, in diabetic patients with the high blood glucose levels, one of three of glucose may pass through the glucose reduction pathway, and this depletes some components and eventually leads to cell damage. And the aldose reductase is the first enzyme in the sorbitol aldose reductase pathway, or shortly polyol pathway, and is responsible for the reduction of glucose to sorbitol as well as the reduction of galactose to galactose. So this is another sugar alcohol. So galactose to dulcitol or galactose, catalyzed by the aldose reductase. And too much sorbitol and also dulcitol trapped in retina cells, lens cells, Schwann cells that myelinate peripheral nerves is a common consequence of prolonged hyperglycemia accompanying poorly controlled diabetes. And this can damage these cells leading to retinopathy, cataracts, and peripheral neuropathy. And glycerol is another sugar alcohol of glyceraldehyde and dihydroxyacetone, the simplest monosaccharide. And glyceraldehyde is a aldotreose and dihydroxyacetone is a ketotreose, and the glycerol is the backbone of triglycerides and glycerophospholipids. Mannitol, the sugar alcohol of mannose, and remember the mannose is the isomer, is an epimer of glucose. So also mannitol is also an epimer or isomer of sorbitol. While they have a similar structure, actually they have very different sources, melting points, and also uses in nature. Mannitol is used, for example, after head trauma to lower acutely elevated intracranial pressure until more definite treatment is available. It can also be used in some cases of kidney failure with low urine output to reduce pressure in the eye, increase the extraction of certain toxins, and treat fluid retention. It is also used as a flavoring agent, but the most important one is as a therapeutic agent. Ribitol or adonitol, the sugar alcohol of ribose, is a crystalline pentose alcohol formed by the reduction of ribose. It occurs naturally in the plant Adonis vernalis and in the cell walls of some gram-positive bacteria in the form of ribitol phosphate in teichoic acids. It also forms part of the chemical structure of the riboflavin. This is the most important, this is the significance of the ribitol to be a part of riboflavin, vitamin B12, and as well as its coenzyme flavin mononucleotide, flavin FMN, mononucleotide, a nucleotide coenzyme used by many enzymes in metabolism in organism, and these are called flavoproteins. And erythritol is an organic compound that is naturally occurring the carbon sugar alcohol of the erythrose. It is used as a food additive and sugar substitute. And it is synthesized from corn using enzymes and fermentation. And erythritol is 60 to 70% as sweet as sucrose, which is the table sugar. However, erythritol is almost completely calorie-free and does not affect blood sugar or cause tooth decay. And sugar acids. Sugar acids are oxidation products of monosaccharides. They are very important biologically. And there are three types of sugar acids: aldonic acid, saccaric, and uronic acids. Aldonic acids, for the aldonic acids, how they occur. The aldehyde group at the first carbon of an aldose is oxidized and changes to carboxyl group, so first carbon, or an aldose, which carry the carbonyl carbon. So in general, if it's an hexose, we call them hexonic acid. If it is glucose, glyconic acid. So onic acid, galactose, galactonic acid, mannose, mannonic acid. And glyconic acid is an important intermediate metabolite in the synthesis of pentoses in hexose monophosphate shunt or pentose phosphate pathway. An oxidation of the carbonyl carbon of glucose to the carboxyl level, which produces the glyconic acid, is important. It is also used in medicine as a harmless counteragent to which positively charged drugs such as quinone or ions such as calcium ions can be administered. The saccaric acids or the aldaric acid, it occurs when the aldehyde groups at carbon 1 and the primary alcohol groups at carbon 6 of aldose are oxidized to carboxyl group. So they have two carbon atoms oxidized to carboxyl group in aldaric acids. And the oxidation product of glucose is glucaric acid, mannose, mannaric acid, and etc. They have no biological significance, at least till now. And the last group is uronic acids. They are formed when only the primary alcohol group of an aldose is oxidized to carboxyl group. This means the six carbon for a glucose, galactose, and mannose. And in general, hexuronic acid is the oxidation product of hexoses. And the most common is glucuronic acid, which is the oxidation product of glucose, which converts this oxidation converts this alcoholic group in the six carbon, which is primary alcoholic group, to the carboxyl group. It is found in the structure of glycoproteins and mucopolysaccharides. This is the formal name. Nowadays, we prefer to say glycosylaminoglycans. And it has function in the detoxification processes and the removal of toxic substances. And both aldonic and uronic acids form stable intermolecular esters called lactones. And here you see the uronic acid of glucose, which is glucuronic acid, but it's in the ionized state. The carboxyl group in aqueous media donate the proton, which makes a conjugate base. The N is in the beta form and also in the D form, so beta D-glucuronate. This is the uronic acid of glucose, and this is the aldonic acid of the glucose, D-gluconate, and here the carboxyl group. And again, it's done as the protein aqueous media, so it exists in the form of the gluconate and the glucuronate. And phosphate esters. And their intermediates in the synthesis and metabolism of carbohydrates are usually not the monosaccharides themselves, but their phosphorylated derivatives. The esterification of phosphoric acid with one of the hydroxyl groups of a monosaccharide forms a phosphate ester, as in glucose 6-phosphate, and the first metabolite in the pathway by which most microorganisms oxidize glucose for energy. Sugar phosphates are relatively stable at neutral pH and they carry a negative charge. One effect of monosaccharide phosphorylation in cell is to trap the monosaccharide inside the cell. Most cells lack plasma membrane transporters for phosphorylated monosaccharides. Phosphorylation also activates monosaccharides for subsequent chemical transformations. Several important phosphorylated derivatives of monosaccharides are components of nucleotides. So they are so important monosaccharide derivatives. Here's the two monosaccharides, glucose and fructose, and their phosphate esters or their phosphate derivatives. And for the glucose, there are two given here: glucose 1-phosphate, glucose 6-phosphate, and for fructose, fructose 6-phosphate, and fructose 1,6-B-phosphate. And also there are common names: Corey ester, Robinson Nibbles, and Hardy-Young ester, and they are important intermediate metabolites in carbohydrate metabolism. And we'll finish the monosaccharide derivative. And now the title is Disaccharides: Glycosidic Bond, Maltose, Cellobiose, Lactose, Sucrose, and Trehalose. Carbohydrates formed by condensation of two cyclic monosaccharide molecules are called disaccharides. And look, two cyclic monosaccharides. This means for the cyclic monosaccharide structure, you should remember power projection. 1 mole of water is released during this process. And the general formula is n number carbon and n minus 1 number of water molecule, because 1 mole of water is released during this process. During the formation of disaccharides, monosaccharides are linked by glycosidic bonds or linkage. The oxygen bridge formed when two monosaccharides are joined is called a glycosidic bond, and it's in the form of O-glycosidic bond. Disaccharides consist of two monosaccharides covalently linked by an O-glycosidic bond, formed by the reaction of the hydroxyl group of one monosaccharide molecule, typically cyclic, with the anomeric carbon of the other. And this reaction represents the formation of a hemiacetal and an acetal form of an alcohol in the form of one monosaccharide gives carbonic carbon, and the second one gives it alcohol group. The compound is called glycoside, and if the glycoside bonds or if the glycoside occurs between two monosaccharides, we call it disaccharides. There are also N-type glycosidic bonds, but it occurs in glycoproteins and nucleotides. Two monosaccharide molecules can form two different glycosidic bonds. And the one is maltose type bond. It occurs when the carbonyl group, which I mean the aldehyde or ketone in Hubble projection hemiacetal structure, of one monosaccharide bonds with the alcohol group of another monosaccharide. And the second type is Trehalose type bond. It occurs when the carbonyl group, aldehyde or ketone group, or the hemiacetal part of one monosaccharide molecule bonds with the carbonyl group, aldehyde or ketone or hemiacetal, of another monosaccharide molecule. Since the aldehyde or ketone group of at least one of the monosaccharides is free in maltose type bonding or linkage, the saccharides with this type of linkage have reducing properties because they still carry a free hemiacetal structure. Disaccharides with trehalose type linkage do not have reducing properties since the carbonyl groups of both monosaccharides are used for the linkage. So they both have the acetal structure. Alpha glycosidic linkage or bond and beta glycosidic bond or linkage. So the alpha and beta, so this is designations for the anomers. Remember the anomers, there are two anomers of glucose, alpha anomer and beta anomer. So depending on which donate its carbon to the bond formation, the glycosidic bond type varies. So there are two types, alpha and beta. So remember, according to the configuration of the hydroxyl groups in the anomeric carbon, there are two forms, alpha and beta anomers. Thus, we have alpha glucose, beta glucose, alpha galactose, beta galactose. If the hydroxyl group of the alpha anomer is used in the glycosidic bond, it becomes alpha glycosidic bond. If the hydroxyl group of the beta monosaccharide, which I mean the beta anomer, is used, it becomes a beta glycosidic bond. And remember, and pay attention, because here the hydroxyl group, I don't mean the alcoholic group. I mean the hemiacetal structure, the hydroxyl group in the hemiacetal structure, not the alcoholic groups. This is so important. When showing the glycosidic bond, the carbon atoms between which the bond is realized are also shown. If the link is between the hydroxyl group at carbon 1 of the alpha monosaccharide and the secondary alcohol at the carbon 4 of the monosaccharide, this link is shown as the alpha semicolon 1, represent this one, giving the alpha anomer to the bond formation, and arrow indicates the direction to the second monosaccharide, which donates its 4-carbon alcoholic group for the glycosidic linkage. Thus, the name will be alpha 1,4 glycosidic bond or glycosidic linkage. The alpha and beta form of the linkage is of great importance because different disaccharides are formed depending on whether the connection between the same monosaccharides and the same carbon atoms is alpha or beta. And the good example is maltose and cellobiose. Even they carry the same type of monosaccharides, the different glycosidic linkage, different pattern of glycosidic linkage forms them. Thus, it provides a different characteristic, or it provides the disaccharides having different characteristics and different molecular shape. And here is in my opinion, a good table, a perfect table for you to study the disaccharides, their monosaccharide units, and the glycosidic bonds. And look at the table for 5 seconds to realize an interesting thing. And yes, look at the monosaccharides. Look at the number of the glucoses here. Glucose, lactose here, glucose, again glucose, and glucose. Glucose is everywhere. Another example that to say why glucose is so important, why the glucose has a central role in carbohydrate metabolism, plus in our metabolism, plus in mammals' metabolism. Glucose is everywhere. And despite being glucose is component of all disaccharides, there are also some other ones, as in lactose, galactose, and sucrose, fructose. But look at here, the maltose, cellobiose, and the trehalose all consist of only glucose in different anomers, but they have different bonding pattern, different type of glycosidic bonds. This is the importance. This is the thing that they are different monosaccharides. And even if you compare the alpha maltose and the trehalose, look at the components. Alpha glucose, only alpha glucose. But look at the glycosidic bond in alpha maltose, alpha 1,4, but in trehalose, alpha 1,1. And compare these two. The same building pattern, alpha glucose and beta glucose, yield different disaccharides due to different pattern of glycosidic bond, alpha 1,4 and beta 1,4. So the type of bonding is of great importance. And now let's discuss them one by one. Maltose. Maltose is formed when two glucose molecules are linked by alpha 1,4 glycosidic bonds. Both glucose molecules can be alpha glucose, or one can be alpha glucose and other beta glucose. And as a result, there are two forms, alpha and beta maltose. Maltose is also called malt sugar. It is of plant origin. It is a plant sugar, and also it's a component of starch. It's a disaccharide unit of starch. And alpha maltose formed by the condensation of two molecules of alpha glucose, and beta maltose occurs due to condensation of one molecule of alpha glucose and one molecule of beta glucose. So the difference in glucose anomer creates the beta maltose. But in both maltoses, the bonding pattern is same, alpha 1,4, and it has a reducing property. Let's look at here. We have the alpha D glucose and alpha D glucose, and there are two possibilities here. It may be alpha maltose or trehalose, and the bonding type determines this. And here the bonding type is alpha 1,4 glycosidic bond. And here you see 1 mole of water is removed during the condensation reaction. And at the end, an oxygen bridge occurs between alpha D glucose and alpha D glucose. And also remember, here actually I omit to say glucopyranosyl because they are in cyclic structure. And in the nomenclature of cyclic structure, actually we need to represent the type of ring, because it may be either pyranosyl ring or the pyranosyl ring or furanose ring. And it is here the glucose is in the pyranose. So actually this is the glucopyranose. And also this one, alpha D glucopyranose. Why we call it pyranose? Why we call it alpha? Because the hydroxyl group in the anomeric carbon in the hemiacetal structure is at the right side. Okay, and these red arrows indicate whether it's alpha or beta, the side of the hydroxyl group. And this is alpha maltose. In trehalose, the bonds occur here, this one and this one, alpha 1,1. And this is the Harvard projection, the motor projection of Harvard. And remember how we determine it is the glucose. We should check the hydroxyl groups at the third position in the upper part, and the fissure is at the left side, the rest in the right side. And this is the alpha anomer because the hydroxyl group in the acetal structure below, and also this is in the below. This is alpha D glucopyranose, alpha D glucopyranose, and this is alpha 1,4 glycosidic bond. And the fourth carbon here, there is an hydroxyl group, alcoholic group, the secondary alcoholic group. So alpha 1,4 glycosidic bond. And also we say in detail the name of the alpha maltose depends on the type of the monosaccharide units. It will be alpha D glycopyrenosyl 1,4-alpha-D-glucopyranose. And yes, here, look at, we have alpha-D-glucopyranose and beta-D-glucopyranose. But what I said, we can omit the term pyranose. Thus, I wrote to be simple here glucose instead of glucopyranose, but we know they are in the pyranose structure. So there are the two possibilities that these two monosaccharides can give, which one is the beta maltose and the another one is cellobiose. And who determines this? Yes, alpha 1 type of glycosidic bond or the pattern of glycosidic bond. And here it is the alpha 1 for glycosidic bond, thus it is a beta maltose. Because in cellobiose it will be beta 1 4. So the beta glucose will donate it a hydroxy group in the acetal structure. And look at here, it is the left side, that's beta, here right side, alpha. And this is the Hubble projection. And again, to identify whether it is galactose, glucose, or mannose, focus on the third, fourth, and the second. And the third carbon atom, the hydroxy group at the upper part, only the rest in the right, the below. So this means they are the right part in right direction in the Fisher projection. This is glucose, also this is glucose. This is the first and hemiacetal structure here, but this is a full acetal structure. But when we look at the molecule in total, due to there is a free acetal group, that's a free hydroxy group, it has reducing feature, alpha 1 4, and this is beta maltose. Cellobiose. Cellobiose is a disaccharide formed by linking two glucose molecules with a beta 1,4 glycosidic bond. No, beta 1,4. And it is composed of an alpha glucose and a beta glucose. That means its components, or its monosaccharide units, are same with the beta maltose, but the bonding pattern is different. Beta 1,4 glycosidic bond. It is of plant origin and is not found free in nature. It is a disaccharide unit of cellulose polysaccharide. It shows reducing properties since alpha glucose in its structure has a free anomeric carbon, which means hemiacetal structure. And yeah, as you see, the alpha D-glucopyranose and the beta D-glucopyranose forms the cellobiose with beta 1,4. And alpha 1,4 gives beta maltose. Beta 1,4 gives cellobiose. And here is the Hubble projection from one here, the hemiacetal. It's a free. This is the acetal, but due to a free group in a molecule, in total of the molecule, it has a reducing feature. Lactose. It is also called milk sugar because it is found only in milk in nature. This means it is synthesized from mammary glands. If too much lactose is produced or milk is not expressed fast enough, it passes into the bloodstream. The organism cannot use the unhydrolyzed disaccharide. This is general for all disaccharides and also for lactose. And unused lactose is excreted in the urine. This is called lactosiuria. It is found in milk at a rate of 5%. Various microorganisms in milk can convert lactose into lactic acid. This causes the milk to turn sour and curdle when boiled. And it is one of the two saccharides found freely in nature together with sucrose. Lactose is composed of 1 mole of glucose and 1 mole of galactose monosaccharides, and the bonding pattern is beta 1,4 glycosidic bond. And there are two forms of lactose, alpha and beta lactose. And so the alpha and beta, I remember, comes from the anomerism. This means the type of which anomeric hexose they carry, the difference in lactose happens. As a common feature, both lactoses carry beta anomer of galactose. So beta galactose. Depending on whether glucose is alpha or beta anomer, alpha lactose and beta lactose are formed. And it has a reducing property. So this means lactose, cellobiose, and maltose, they all carry maltose type bonding. Yes, here the galactose is in pyranose again. Actually, this is beta D-galactopyranose. And here the alpha D-glucopyranose. And the bond is always same. So always the beta galactose donates its hydroxyl group to the bond formation. And glucose donates its alcoholic group at the fourth carbon, secondary alcoholic group, removing water, which makes it the beta 1,4-glycosidic bond. And we have the alpha lactose. We call it alpha lactose because it has an alpha glucose. We know the galactose is always beta form. And here is the Hubble projection of alpha lactose having alpha D-glucopyranose and beta D-galactopyranose. And the name actually is the beta D-galactopyranosyl 1,4-alpha D-glucopyranose. And how we determine or how we determine whether or recognize this is the glucose and this is the galactose. Let me remind you, focus on the third carbon, second carbon, and the fourth carbon for the differentiation of glucose, galactose, and mannose. And remember, glucose and malactose are epimers, mannose and glucose are epimers. And here only the third carbon carry the hydroxyl group at the upper part, but in the galactose 3 and 4, of course, at the upper part, both 3, 4. So remember this projection. Something can simplify. If it is 3, this is glucose in galactose 3, 4. And then rotate to the clockwise. So the 3 and 4 will be the upper part. And here the beta galactose donates its hydroxyl group at the hemiacetal structure, which turns into full acetal, the alcoholic group. Due to the lactose carry and free hemiacetal in the glucose moiety, it has reducing feature. So the bonding pattern is same in the beta lactose, but the difference is in the glucose. It's in beta anomer. In beta anomer. And here is the projection. The path forms the beta lactose is same as you see here. The difference in the beta glucose here, and it has reducing feature. Sucrose. It is also called table sugar, also known as saccharose and cane sugar. It's a disaccharide formed by alpha D-glucose and beta D-fructose, linked by a glycosidic bond. It is of plant origin. It is mostly found in sugarcane and beet in nature. And it is found in most fruits and candies. It's a very sweet substance. It's a very sweet sugar, disaccharide. Since it does not have free aldehyde and ketone group, which I mean there's no hemiacetal structure in the sucrose, it does not have reducing properties. So there is no free anomer, no hemiacetal structure. It's all full acetal structure. Hydrolysis of sucrose by acids occurs faster than all other disaccharides. Lactose has an acid hydrolysis rate of 100, maltose 127, but sucrose 1240. Sucrose normally turns polarized light into right direction, 66.5 degree, but after it's hydrolyzed by a special enzyme called saccharase, it turns polarized light to the left at an angle of 39.5. And this is due to the fact that all the glucose and fructose produced after the hydrolysis of sucrose. Or this means the hydrolyze will yield the monosaccharides. If heated to 200 degrees Celsius, it loses water and becomes a brown amorphous mass. This is called caramel. It can only be used by the body when given orally. Sucrose and glucose are the most important sugars in our food, in human food. And the sucrose is a source of glucose and fructose. The body easily absorbs the monosaccharides of hydrolyzed sucrose and uses them for various purposes, including energy production. When disaccharides, lactose, sucrose, are given directly into the bloodstream, the body cannot utilize them, and they are excreted in the urine like a foreign substance. And here's the formula of demonstration of how bonds occur with alpha D glucose is the pyranose form, glucopyranose, and fructose is in furanose. So this is beta D fructofuranose, and so both donates their acetal hemiacetal structures to form the full acetal structure. Thus, they have no reducing feature. So the glycosidic bond, it is shown as either glucose alpha 1 beta 2 fructose or fructose beta 2 alpha 1 glucose. So this is sucrose. And this is have a projection of this is the glucose in pyranose, glucopyranose, and fructose in furanose, fructofuranose structure. And here is the full acetal parts, and there is no any hemiacetal free hydroxy group that has no reducing feature. Trehalose, it consists of two alpha D glucose units linked to each other by alpha 1 glycosidic bond. So we know the glucose has an anomeric carbon at the first position, so both donate, thus no reductive. And it's found in the hemolymph of many insects. And here the formula showing the alpha 1 glycosidic bond between two alpha D glucopyranose. And we finish the disaccharides. Yeah, we have reached the last title of the topic, oligosaccharides. Oligosaccharides are short polymers consisting of several monosaccharides joined by glycosidic bonds. In other words, oligosaccharides consist of short chains of monosaccharide units or residues joined by characteristic links called glycosidic bonds. And at one end of the chain, the reducing end, is a monosaccharide unit whose anomeric carbon is not involved in a glycosidic bond. In cells, most oligosaccharides consisting of 3 or more units do not occur as free carbohydrates but participate in the structure of glycoconjugates, and these are the glycoproteins and glycolipids. Glycoproteins are branch and structurally diverse carbohydrate-protein conjugates. About half of all proteins in mammals are glycosylated, and about 1% of all mammalian genes encode enzymes involved in the synthesis and binding of these oligosaccharide chains. The biological advantages of adding oligosaccharides to proteins are slowly emerging. Very hydrophilic carbohydrate clusters alter the polarity and solubility of the proteins they are conjugated to. Oligosaccharide chains bound to newly synthesized proteins in the endoplasmic reticulum and elaborated in the Golgi complex serve as target tags and also play a role in protein quality control, targeting misfolded proteins for degradation. Glycoproteins are not the only cellular components that carry complex oligosaccharide chain. Some lipids also have covalently linked oligosaccharides, and they are the glycolipids. Gangliosides are membrane lipids of eukaryotic cells, and the polar head group, the part of the lipid that forms the outer surface of the membrane, is a complex oligosaccharide containing a sialic acid and other monosaccharide residues. And so this means the gangliosides are a type of glycolipids. And some of the oligosaccharides moieties of gangliosides, for example, those that determine human blood groups, are identical to those found in certain glycoproteins and therefore also contribute to blood group type. Like the oligosaccharide moieties of glycoproteins, the membrane lipid moieties are usually, perhaps always, found on the outer surface of the plasma membrane. Glycoproteins are branch and structurally diverse carbohydrate protein conjugates. The carbohydrate is linked to its anomeric carbon by a glycosidic bond to the hydroxyl group of serine or threonine residue, as seen here. Serine and threonine, which they carry hydroxyl group, and they, as in here, the serine with the O-link glycosidic bondage or glycosidic linkage. Or, as in here, the asparagine, another amino acid, which is carry an amide group. So this, and in this oligosaccharide protein conjugation, the bond type is N-glycosidic bondage or glycosidic linkage. And some glycoproteins have a single oligosaccharide chain, but many have multiple oligosaccharide chains. Carbohydrate can make up 1% to 70% or more of the glycoprotein by mass. And as a final slide of this topic, I want to talk about some three oligosaccharides in nature. Raffinose, a trisaccharide composed of galactose, glucose, and fructose. And it is the most abundant oligosaccharide in plants after sucrose. Maltose consists of glucose, fructose, glucose, a non-reducing trisaccharide synthesized by many sap-drinking insects. It is found in plant sap and especially in pine trees. It is involved in reducing the stress caused by osmosis in insects, and also it is a food source for bees. And Stachyose, a tetrasaccharide composed of galactose, glucose, and fructose. And actually, another word also we can say it's a galactose derivative of this moiety, galactose, glucose, fructose. This is raffinose. It is the galactose derivative of raffinose, or it is formed by attaching one more mole of galactose molecule to the raffinose. It's a tetrasaccharide found in plants. And also there are other ones in nature, such as xanthaniose, verbascose, etc. Okay, my dear friends, we finished the carbohydrates part 2. And here is a reference of the topic, and also for you for further reading and study. And the next topic is Carbohydrates-III. And for more about biochemistry, clinical biochemistry, and laboratory science, etc., you can visit my website, BiyokimyaVET, and also you can follow my Instagram account, BiyokimyaVET. And don't forget to subscribe to the YouTube channel, BiyokimyaVET, to like the video, this is so important to reach more people, and turn on notifications to stay up to date. See you in our next topic, my dear friends. Goodbye.